Hello there. I want to talk about our next unit on cells. And the first topic we usually discuss in our unit on cells is the early discovery of the cell. So some history tied in with discovery of certain things about cells. All right, so there are some things that you may already know about cells, and if we were together, we would brainstorm these ideas and share them. But individually, try to think of some things that you already know about cells. And the history part of today's notes, they're all going to be white old men that contributed to the formation of what's called the cell theory. And they're all also European because most of these contributions were before America was America. Okay, so the first person that we're going to talk about, if you see that date there, is 1665. A scientist by the name of Robert Hooke. You don't need to know dates, but you do need to know their contributions. So Hooke, you can see an image of him there with his wig on. He was a scientist and he had a decent microscope. And what he was doing with his microscope was that he was looking at cork cells. And if you know anything about cork, it's dead plant tissue. It's been dehydrated. And so when he was looking at these cork cells underneath the microscope, he saw those little chambers or boxes. And he came up with the term cell that we use. He coined that term cell because it reminded him of empty chambers that monks sleep in. And so he is the first person we give credit to for developing the word or coming up with the word of cell. Next, not too long after Hooke, I think Leeuwenhoek and Hooke actually kind of were frenemies, so to speak. It was a Dutchman by the name of Anton van Leeuwenhoek. He's known as the father of the modern microscope, and you can see his engineered microscope there, so it was a little bit more powerful than what Hooke was using. And he is given credit as the first person to observe bacteria or microorganisms. So what he did was he had collected pond water, and with the use of his microscope, he saw various bacterial cells from that pond water when he was using his microscope. We also give him credit for the first person to observe living cells. Hooke was looking at dead cells in order to create the cork and to dehydrate the plant cells so they're dead. And so not only is Leeuwenhoek the first person to observe bacterial cells, but he's also the first person to observe living cells. Almost a century later, we're going to look at three German scientists who helped contribute to what's called the cell theory. In 1838, Matthias Schleiden, he was German, concluded that all plants are made of cells. He was a botanist, so he was studying plants. Very close to Schleiden was Schwann. Schwann was a zoologist, so he studies animals, and he also concluded that all animals are made of cells. And then lastly, a physician, he was a medical doctor, Rudolf Virchow, concluded that life has to come from pre-existing cells. So he did research on cancerous cells, which we know that cancerous cells actively divide and reproduce rapidly. And so he concluded that cells must come from pre-existing cells. Which leads us to our three parts of the cell theory. This is still used today. It is a theory. So remember, it can change if new evidence were to come out. But there are three parts of the cell theory, and one is that all living things are composed of cells. Two, that the cell is the basic unit of structure and function, and that three, cells come from already existing or pre-existing cells. When we classify cells, there are different ways we can classify them. For right now, we classify them as prokaryotic or eukaryotic. Prokaryotic means that they do not have a membrane-bound nucleus inside of them, so their DNA, their genetic material, is not contained within a structure or a nucleus. So examples of these types of cells are bacterial cells. They are prokaryotic because their genetic material is not contained inside a nucleus. Eukaryotes are the opposite. The term eukaryote means true kernel, so they do have a membrane-bound nucleus and kernel like a seed. not like a captain or admiral or something like that. So eukaryotics, like we are, their DNA is contained within a true membrane-bound nucleus. And membrane-bound means that it's surrounded by a membrane. So if we're thinking about classification, Animalia, Plantae, Protista, or protists, and fungi are all eukaryotic because of that membrane-bound nucleus. However, despite having that key difference, there are some similarities. Both prokaryotes and eukaryotes have a cell membrane, which we can also call the plasma membrane. And just wrapping up that chapter, remember we see that phospholipid. We also see proteins and carbohydrates as well within cell membranes. Remember, all living things have genetic material in the form of DNA, and that would be CHOMP. So we would see that nucleotide of nucleic acids. All prokaryotes and eukaryotes have the organelle of ribosomes. Ribosomes do not have a membrane surrounding them, and so they're found in all types of cells. And then, of course, cytoplasm, that fluid within cells. So you can see two images of prokaryotic cells and eukaryotic cells. And typically, eukaryotic cells are going to be more complex because they have more internal structures, and they're also going to be larger in size as well. And that's it. I'll see you later.